Okay, hello. My name is Hedrick Quintana. I'm a Panamanian medical doctor. I am doing a master in science in infectious disease control in Södertörns högskola in Sweden. I have to defend my thesis the next month. Okay, Bionicdance is a cartoonist, and he tried to put aside the Draw Mohammed drama in order to highlight the importance of the discovery of life, of artificial life made by Craig Venter. She was not able to see what's going on because she lacks of the vocabulary needed, and she's right. For instance, Einstein made a layman version of the special theory of relativity in order to make the public aware about his discoveries. Richard Muller is a PhD in Berkeley University, make a special videos of physics for dummies he called Physics for Presidents, and all the links of this special physics guide are below in the description. In this video, I am trying to translate into a layman words what is going on with this new discovery. I could have some mistakes, I admit it, but I am pretty sure they are not intentional. The first issue to understand what is going on here, you have to know exactly what is the nature and what other researchers have done before this researcher start with the experiments. The first thing that you should know is the cells are tiny computers. The information to run them came from the parents of these cells, and this information is called genome. The genomes encodes the order or some building block of life called amino acids in order to construct proteins. These proteins per se or by chemical reaction that are impossible to do at the temperature and the pressure within the cells define the physical feature of each organism. The genome in some viruses like hepatitis B, herpes, the shingles viruses, and all living cells is codified in a large molecule called DNA or deoxyribonucleic acid. The other viruses like influenza, HIV, hepatitis C are codified in another slightly different molecules called RNA or ribonucleic acid instead. Some viruses are capable to mix or integrate the whole genome in another species, or especially the target species that they have. Cellular viruses and hepatitis C, as I told you before, are not able to do such integration process. But viruses like HIV, they have special mechanisms you can see in the picture, and they integrate their genome within the host. The DNA is packed in several units called chromosomes. If the cell has a unique chromosome and it doesn't have any border with the other parts of the cell, this is a bacterium or a prokaryote. If the cell has several chromosomes, more than one, and is bordered by a membrane, it is an eukaryote, and it could be a plant, an animal, or some unicellular living forms like yeast, mold, malaria parasite, for instance, and worms, whatever. The other aspect that usually is not in the nature, but the researcher before this team have done is called the molecular cloning. The molecular cloning is when you incorporate foreign protein from a species to another one. The complete genome has only been transferred from a cell from one type of cell in a species to the same animal species, but with another type cell. A classical example of this molecular cloning could be the vaccine against hepatitis B. In that case, the viral product is produced when it's introduced in a bacterium Escherichia coli, sorry, in English, uh, in order to produce safety large amount of viral protein with a very low cost. And this is used to get a coverage in a very safe, very large population of people. Okay, let's see what was really this guy have done. The first thing is they took the chromosome of a living creature called Mycoplasma genitalium, that is a human pathogen that infects, as the name says, the genitals, and also can infect the respiratory tract. At the time that they started to work with that, at least in 2002, you have the smallest genome known. They start to delete all the genes that are not vital for the survival of this organism, and put these genes into another cells. The recipient cells is expressing this new genome that they have developed, and there's alive, the cell. They need a lot of time killing their own neurons, and also computers to help in order to develop this new adventure. Also, they put this new genome, that is a bacterial genome, into a yeast that's, as I mentioned before, is an eukaryote. Why they do that? Because the bacteria has is something very, it's not common in that kind of bacteria, but they have a very low growth rate, and they need to have very rapidly get new fresh information with the minimum amount of mistakes. And usually this kind of yeast can help in that case. This is a breakthrough or new discovery that this was not done before. Also, they start to write down some watermarks with the email, the address of the special of the genome, also the name of the sponsors that paid for this kind of experiments in non-vital areas of this new genome. The idea is to say, fuck yeah, we did it before anyone else. Okay, I think I have covered the most important part of this of the experiment. I would like to read some comments in order to know if this was clear or this was some messy, but I hope to be the most clear as possible. Also, I added some links in the description with some weird terms that I use, like genome, DNA, whatever. Also, the Wikipedia page of the Mycobacterium laboratorium, that was the name of this new bacteria that they developed, and also the link of the Bionicdance runs, and also the links of the briefing that was done by Craig Venter and their team. Okay, thank you everybody.